Robert. Tusan. Robert, hello.
Sí, estoy aquí.
Ok. ¿Querés que te presente nuevamente?
No escuché nada por eso.
Ok. No veo nada. Ahí vamos.
Hola.
Hola. He is Robert Satorre. He was born here in Argentina. He is a cognitive neuroscience at the Montreal Neurological Institute of McGill University. His research explored the functional and structural organization of the human brain, with a special emphasis on two complex and characteristically human abilities, speech and music. He and his collaborators have published over 300 scientific papers on topics including structure and function of the human auditory cortex, pitch perception, auditory spatial processing. His talk entitles From Perception to Pleasure, the Neuroscience of Music and Why We Love It. Thank you, Robert.
Okay. Okay, can you see my screen?
Yes, we see it.
Excellent. Buenos días. Muchas gracias por la invitación. Thank you very much. It's very nice to be able to talk to you. I wish I were there, but Zoom is the next best thing. And so today I'm going to talk to you a little bit about some of the research that we have done over the years, trying to understand music and the pleasure that we derive from it and the neural basis for that phenomenon. Now, we know that music is really very ancient in the human species. If you look at archaeological results, we find that already 40,000 years ago, humans were making beautiful musical instruments like this flute, which was found in what is now Germany. So this means that music is not only ancient, but also must be very important for humanity, because at the time this instrument was made, life could not have been easy in the Upper Paleolithic period. And yet people were spending effort and resources to create music. So it's sort of part of what I like to call our human mental machinery. It's part of our original endowment as humans, part of our basic cognition. And the question is, why might this be? And the most famous quote by Charles Darwin about music is this one, where he said, "Neither the enjoyment nor the capacity of producing musical notes are faculties of the least direct use to man, and therefore they must be ranked amongst the most mysterious with which he is endowed." In other words, he could not figure out why music exists. He did not really have a good idea. That's what he said in The Descent of Man in 1871. But 10 years later, he published his autobiography. And as you may know, Darwin was late in his life was rather depressed and unhappy. His daughter had died, and he was suffering from what we might today call anhedonia. And what he wrote in his autobiography is this: "If I had to live my life again, I would have made it a rule to read some poetry and listen to some music at least once every week, for perhaps the parts of my brain now atrophied would have been kept active through use," which I think is a remarkable statement. And then he went on to say that the loss of these tastes is a loss of happiness and may possibly be injurious to the intellect and to the moral character by enfeebling the emotional part of our nature. And in a way, I think what he's saying here, when he's just reflecting, personally, he's sort of answering the question that he couldn't answer 10 years earlier. He's basically saying that music and other art forms are essentially about strengthening the emotional part of our human nature. So from that, we can conclude that music has a lot to do with emotional arousal. It has to do with the expression and the communication of emotions and with the regulation of emotional states, either from one individual to another, but also within ourselves. And so the question that I want to try to understand is how this happens in the brain. Why and how is it that music can cause us pleasure and generate emotions? And the answer I will give is based on these 3 points. First of all, I think we need to understand the auditory cortex and its circuitry, because that's where we have the neural mechanisms that permit the perception and the production of music, as well as prediction mechanisms that I'll get to. The second mechanism has to do with pleasure and reward prediction, and that's a separate set of structures, the striatum, for example. I'll show you in the next slide. And my main thesis is that it's the interactions between these systems that are the source of musical pleasure and more generally of aesthetic pleasure. So these 2 systems, here they are in a diagrammatic form. We can consider the first one here on the left to be consisting of mostly cortical interactions, so the auditory cortex and its connections to the rest of the brain. And then the second system is the reward system, which is found mostly in deep structures, mostly subcortical structures, but also including some cortical regions, as shown in this mid-sagittal view of the right hemisphere. So let's take this apart. I'm going to start with the auditory system. And as you probably know, auditory inputs reach the cortex via a series of brainstem nuclei that I won't talk about today, but they're shown here, brainstem and midbrain and thalamic nuclei. And then in the cortex, the auditory cortex is found in the superior temporal plane. So if we open up the Sylvian fissure here, we see primary regions mostly here in orange, and then there are additional areas, both anterior and posterior, to the primary region. And these regions have many different important and interesting functions. I won't really have time to tell you very much about them, except I want to mention one particular function, which I think is really essential for music, and that has to do with the encoding of periodicity, frequency information. And we can investigate this in different ways. In one of the experiments we've done in our lab, carried out by my former student, Emily Coffey, we used magnetoencephalography to look at the so-called frequency following response. So on the top here is an audio sound. This is just a vowel sound, an ah, like that. And what you see in the bottom is the MEG trace, the fluctuation in the activity here, which follows the frequency of the actual stimulus. So this shows us that the auditory system is encoding the frequency content, and therefore that's how we get pitch perception. When we look about where this is coming from, there are generators of this potential in the brainstem nuclei that I mentioned a second ago. Those are very well established. But there are also generators in the auditory cortex, especially on the right side, but it's bilateral, as you can see here. And these signals are interesting because the strength of the signal is related to individual differences in ability to discriminate tones of different frequencies. So the scatter plot shows you for different individuals that the people who have very low frequency thresholds, in other words, are very good at this task, tend to have higher amplitude of this potential compared to those that are worse at it. So this already tells us that your auditory cortex is very important for these very fundamental functions. And there are other functions I could mention as well, but let's leave it there and move to the connections between the auditory cortex and the rest of the brain. I'm going to divide them into 2, dorsal and ventral pathways. And those of you who work in vision may find this very similar to descriptions of the dorsal and ventral visual pathways that were described a long time ago by Leslie Engerleiter at the NIH. So this dorsal region starts sort of in auditory cortex and projects to various portions of the prefrontal cortex, the dorsal premotor cortex, and the parietal cortex. And it's responsible for many different functions. I, again, don't really have a lot of time to tell you very much about them, But one way to conceptualize all of these functions is that it has to do with temporal predictions. So the ability to generate some kind of a model of events as they occur in time. And that also is highly related to the ability to move in time to auditory sequences and to generate auditory sequences. In other words, to produce music, because that has to be done in real time. I'll give you one example of this function, which has to do specifically with metrical organization and therefore is very relevant for music. This is work done by Joyce Chen and Virginia Kenyon a few years ago. And what we did in this experiment was fairly straightforward. We took a simple pattern of sounds, which is shown here in musical notation. And these sounds are very highly metrically organized. Da, 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 da. It's very simple organization. We can take the same sounds and randomly permute them, as shown here. And this is much, much harder to encode. If you look at behavioral data, which I'm not showing you here, people find this much more difficult and make a lot more errors. But it's the same sounds as here. It's just in a different order. So this means that any differences we see between these conditions are not related to the low-level encoding of durations, but rather to the higher-level complex organization of metrical structure. And what we see when we compare these conditions is a recruitment of these dorsal stream areas, in particular the dorsal premotor cortex, shown here, and also some of the prefrontal regions, as well as the auditory cortex. So this means that dorsal stream structures are sensitive to this regularity that's present in the metrically organized pattern, but not in the metrically complex or unmetrical pattern. And therefore, this is evidence for a temporal prediction mechanism. Now we'll move to the ventral stream, the auditory ventral stream, which, unlike the dorsal one, is more time independent. It does not so much rely on events happening in real time, but it tends to accumulate evidence over time periods and therefore generate abstract representations of auditory patterns. And these can be auditory patterns extending over short periods of time, over seconds, but also this ventral stream projects to medial temporal regions, which have to do with memory formation. And in a more general sense, we can conceptualize this ventral stream as having to do with pattern-based predictions. The kinds of protocols that show you engagement of this auditory anterior and ventral areas are, for example, deviance detection. So in this experiment by Mark Schoenwiesner, we see that when you have a pattern of sounds and then you have a deviation unexpectedly from that pattern, you get a response in these regions, which indicates that there is sensitivity in this part of the brain to structural regularities in tonal patterns. These sorts of regularities extend also to long-term memory. So in this experiment from Barbara Tillman, we see a response in the inferior frontal cortex when the chords at the end of this harmonic sequence are relatively more expected or relatively less expected. So the same chords, when they're found in this context, this is a D-flat major chord progression, are considered highly expected, whereas in this context, they're considered less expected. And so again, the ventral stream responds to this relative violation of expectations. This is based on long-term memory because over periods of years, we have heard sequences of this kind, and we know what is expected in a given context. So the auditory cortex and all of the different cortical-cortical connections are essential for almost every aspect of music, not only the encoding of relevant sound features, but the encoding of the relationships between those sound features, also the relevance to motor processes, which allow us to have the ability to generate music, because you couldn't perceive music if someone didn't generate it, obviously. And these same systems are involved in working memory, which I didn't have time to tell you about, And overall, in a broad way, they're sensitive to regularities. Therefore, they are the source of auditory expectancies and prediction. But what does this all have to do with pleasure? Because I said that I was going to explain how we get from perception to pleasure. And so for pleasure, we turn to the reward system, which is shown in this diagram here. It has many components, but I'm going to focus in particular on the striatum, shown here in color, which can be subdivided into more dorsal regions, shown in blue and green, and more ventral regions, shown in red and yellow. This system has been identified for many years now, primarily from animal studies with rodents and also with monkeys, as being the system which is responsible for reward processing, but also other related functions like motivation or salience. And one component of the reward system is also this cortical area, the orbitofrontal and ventromedial prefrontal cortices, which are thought to be involved in valuation, so assigning value to stimuli. Interestingly, these systems are dopaminergic in nature, and Ernest Macerero, who speaks later, will tell you a lot more about the dopaminergic mechanisms. And it's been described by people like Wolfram Schultz that this dopaminergic system is important for reward prediction error. Now, we know that not only in rodents and monkeys, but also in humans, the reward system is active to different kinds of rewarding stimuli. For example, in this meta-analysis from Guillaume Sescus, he was able to demonstrate that with functional imaging, you get very consistent activity in the striatum, as well as in the ventromedial prefrontal region, to monetary rewards. So these are sort of gambling tasks. So money activates the system. Food rewards, these are tasks where people see images of food or sometimes receive something very flavorful, and you get recruitment of this area. And also erotic rewards. So sexual images clearly activate the same system. In fact, they all overlap, as shown in this overlap map over here. Now, these types of rewards you might consider to be biologically important. We need food to survive. We need sex to survive the species, if not necessarily the individual. And money, well, you know, money is fungible, so you can use it to obtain primary rewards. It is not really valuable in itself. And I know for all of you in Argentina, you probably remember back in, I think it was around 2000, when the peso lost its value from one day to the next. So money is not in itself inherently rewarding, right? It's only rewarding because you can use it for something else. Now, music is not like any of these things. It's not directly biologically necessary for survival, and it's not used to buy something else, right? It's just pleasurable by itself. So when we first started doing these studies, we weren't really sure whether music would engage the same reward mechanisms as these more primary or secondary rewards, because music is completely abstract. It is not like these other rewards. But indeed, we found in several different experiments very consistent responses in the striatum to highly pleasurable music. And we've done this over a series of different studies now. The first one of them was done by Anne Blood, who was a postdoc in my lab. And she used, at the time, the brain imaging system that we had available was a relatively low resolution one, positron emission tomography. And we did find the ventral striatum along with other regions. Then 10 years later, with Valerie Salampour, we were able to use a different kind of positron emission system using the radioactive tracer raclopride with carbon-11. And this is interesting because raclopride is a synthetic molecule that binds to dopamine receptors. So we see the striatum here showing dopamine activity. And finally, in 2013, and also subsequent studies that we've done, using functional MRI, we see blood oxygenation response in this region. And I want to tell you a little bit more about this last study, because I think it's very relevant. So in this study, carried out by Valerie Salampour, what we did was we asked people to listen to each of many different musical excerpts lasting 30 seconds. So 1, 2, 3, 4, 5, up to 70 different ones. And each person listened to the music while we scanned them. And then at the end of the 30-second period, we asked them, well, how much money would you be willing to spend to buy that music? So if they really didn't like it, they would say 0. If they liked it a little bit, they might say 99 cents, or they might say $1.29. If they liked it a lot, they would give the maximum amount available, which is $2. And then what we did was we used the amount of money as the regressor on the brain imaging. So the money is a kind of proxy for the value that each person assigned to each piece of music. This allows us to distinguish between individual differences in liking, because not everyone likes everything, and it allows us to give people a lot of different stimuli. And what we found was this image I showed you before, which comes from this analysis. The more money you spend, the more the activity in this region goes up. But that's not the only thing. This shows that this reward system encodes the musical value. But even more interesting was what happened when we analyzed the functional connectivity between the reward system and the rest of the brain. And what we found was that the degree of functional coupling between the reward system and the rest of the brain went up as the value increased. And it went up in very specific regions, particularly in the auditory cortex, very strongly, as well as in other components of the reward system, like the orbitofrontal cortex and the amygdala. So this means that the more you like a particular piece of music, the more the reward system is interacting with the auditory system. And that result is extremely important, I think, because it links together these two systems that we've been talking about, the cortical pathways with the reward pathways. So the model that we've sort of developed is that these cortical systems are responsible for generating sensory prediction errors. So as you're listening to music unfold, you have some internal model of what should happen next, and then different events might happen at different times that are not entirely expected. This sensory prediction error is then propagated via these pathways to the reward system, which generates a reward prediction error, which in turn generates pleasure and emotional arousal, and also leads to learning. Now, this model can be tested in various ways, and actually, Ernest will tell you about this in a little while. But in some work that we did together with Ernest and our colleagues at the University of Barcelona, we identified people who have what we call specific musical anhedonia. And this is very interesting because this population allows us to test the model. So the model stipulates that these two systems aren't interacting when we experience pleasure. Therefore, one prediction of the model is that if we don't experience pleasure, there must be some interruption of this interaction. So, first of all, we identified people who really just don't experience pleasure from music. We call these people musical anhedonics. Importantly, these people do not have any particular disruption of their reward system in general. They enjoy food and money and sex and exercise and drugs, just like most of us do. It's only music that is affected. We contrasted this with the sort of average population who derive pleasure more or less from all of these things. And then we also found a separate subgroup, what we call hyperhedonics to music, who like music above all. When we test them with psychophysiological measures, we see that there is no response in the anhedonics in terms of skin conductance, which is a measure of autonomic nervous system arousal, nor in heart rate as they hear music. Whereas people with average hedonia, as well as the hyperhedonics, of course, have strong modulation of both skin conductance and heart rate. What's really more interesting is when we put people in the scanner, and this is work done by Ernest and one of the students, Noelia Martinez. And what we found here is that as people listen to music in the scanner, if they're anhedonic, you see here the response in red, they show zero or even negative response in the ventral striatum as they hear music. This is not true for the hedonics and hyperhedonics shown in green and blue. Importantly, we had a control condition, which was a monetary task, so a kind of a gambling task that's used very often in neuroeconomics sorts of paradigms. And when we test people with these protocols, we see a perfectly normal response in the people with music anhedonia, shown in the red stripes, compared to the other groups shown in the green and blue stripes. So this confirms that there's nothing really wrong with the functionality of the reward system. When people are playing this gambling game and they gain money, their reward system is active. But it is not active when they hear music. So it's a very interesting dissociation. We can go further than that and look at the functional connectivity. Because remember, according to our model, it's the interaction between the auditory system and the reward system that are responsible for musical pleasure. So to ask that question, we identify a region of interest, shown here in blue, in the auditory cortex, and we look at where the functional coupling is most different across those three groups of musical anhedonics, average hedonics, and hyperhedonics. And we see that the functional connectivity with the right auditory cortex is most altered in the people with musical anhedonia. In fact, the level of connectivity or correlation between the time series of the auditory cortex with the striatum is essentially zero in these groups, or even a little negative on the right side here. The people with average hedonia have a connectivity, as we expect from the previous study I showed you with Valerie Salampour, and the people with hyperhedonia have a slightly elevated level of connectivity. So in other words, the level of connectivity between the auditory cortex and the reward system is predictive of the level of pleasure experienced by those groups, which is in accordance with our model. So we can also then say, well, does this have a structural basis? What happens if we look at the structural connectivity, anatomical connectivity between these different systems? And so remember, I showed you earlier that the auditory cortex connects to the striatum via the orbitofrontal cortex. So therefore, we identified regions of interest here, auditory cortex in blue and orbitofrontal cortex in green. And then we identified the tract that interconnects those two, which is shown in red. And then using diffusion weighted imaging, we look at characteristics of this fiber tract, in particular diffusivity, where a greater diffusivity is associated with less well-organized white matter pathways. And what we see is that the people who score low on a questionnaire, which is designed to evaluate anhedonia, are the people with relatively higher diffusivity here. And this is a different measure of diffusivity, but it's very similar. Whereas people who score high, meaning that they really enjoy music a lot, have relatively low diffusivity, which means a better organized pathway between the auditory cortex and this component of the reward system, which is the orbitofrontal region. So our global conclusion really is that music is a mechanism, I would say, by which humans have learned to enhance or regulate their emotions. And it comes about via the interaction of these two systems. And what's interesting is that the reward system here is phylogenetically very ancient. We share it pretty much with other primates and even rodents. Whereas the cortical system is more phylogenetically advanced, and particularly the frontal cortex, which is the most highly developed in humans compared to other species, is especially important for these predictive mechanisms. And so this is perhaps why music has such power, because on the one hand, it's based on our most cognitively advanced mechanisms for perception, for interactions with the motor system, for memory, and so forth. But at the same time, it's also very much contingent on the activation in these very kind of primitive, if you will, or primal reward system structures, which are phylogenetically ancient. So I will end with that and make sure I thank all of my funding agencies and also all the different people who've worked on this, including some of our colleagues at the University of Barcelona, and some of my former students, like Anne Blood and Valerie Salenpour, and especially Ernest Macerero, who will speak to us a little bit later on and tell you about some very interesting and novel findings that take the story even further. And I thank you very much for your attention. Y cualquier pregunta que puedan tener puede ser en castellano o en inglés. Thank you.
Thank you, Robert. Beautiful talk, beautiful. Ok, ¿alguna pregunta? Sí.
Hello, beautiful, beautiful talk. I just wonder if you think that the functional connectivity that you don't see too well developed in these people that is not having, does not experience a pleasure with the music, if you think that that connectivity is actually built with experience. So all these connections of the reward system with other areas, maybe these people were not exposed to music when they were young. I don't know.
Yes, that's an excellent question. Thank you for that question. It is indeed a little bit of a chicken and egg problem, because we don't know if the lack of connectivity is a cause or an effect. However, when we selected the people to participate in this study, we did, of course, question them very carefully about their early experience with music. And all of them said that, on the one hand, they were exposed to music. They had normal sort of upbringings. They were not deprived in any way. They were not people who, I don't know, grew up in some terrible environment. But at the same time, they also told us, oh, I've never liked music. I don't remember a time when I really liked music. So my impression, and Ernest could possibly comment more on this later on, my impression is that it's probably more a congenital kind of function, if you will. I don't like to call it a disorder, because I think it's just part of neurodiversity. It's not really that there's anything wrong with these people. They just have a different way of deriving pleasure. However, I think it's also possible that both things are true. In other words, it may well be a congenital difference in the anatomical connectivity between auditory and reward systems, but that initial setting may then not develop further, because since these people don't really get much pleasure from music, they're not motivated to ever learn about music, be exposed to music, learn to play an instrument, hang out with friends who play music, etc. So it could well be that an initial difference in the connectivity then leads also to a lack of development with exposure.
Thank you so much. Excellent talk. I also have some, my question was related to Antonia's, that how much the culture modifies these circuits. For example, if you do the same experiment in Brazil or in Rio de Janeiro, something like that, that they have, like they born with music, the babies learn to dance before walking. Would you expect some difference there? And also in a cultural sense, if you repeated these experiments in different cultures, like, I don't know, in India, for example, like, where rhythms are really different, so a different way of perceiving rhythm.
Sure. So a central feature of my model is that you, by exposure, you learn these contingencies of sounds over time, and you learn to generate the predictions, right? And so in that sense, it's entirely culturally based, because the type of sound patterns to which you are exposed will depend on where you grew up and who your parents are and what your culture is. And so indeed, we know, for example, that from experiments that other labs have carried out, that detection of an unexpected deviant is very much tied to the type of music that you are familiar with. So if you play somebody from a Western culture music from India with a wrong note, they may not notice, but someone from the Indian culture, of course, would immediately notice. And vice versa, if the person from India has not been exposed to a certain Western musical pattern, then they won't detect it. So it's very much tied to that. And I do think there is also evidence that the earlier the exposure is to music, the more these patterns accumulate and become strengthened. We know that in general, statistical learning is a very early feature. It's found in infancy, as shown, for example, by Jenny Safran many years ago in the speech domain, but also in the music domain. This has also been demonstrated. So even infants are sensitive to these patterns. And if you don't get exposed early to some of those patterns, it's much harder to learn them. So, for example, Laurel Trainer has done some experiments showing that early exposure to rhythms that are used in the Balkans in Eastern Europe, which are very unusual for people in Western Europe because they're not necessarily evenly divided. You might have a pattern of, 2 plus 3 plus 5 or something like that in the metrical organization. Those are very difficult to learn when you're an adult. It's not impossible, but it's much harder. But if you're exposed to those rhythms very early on in life, you will develop those internal representations of those rhythms, and that makes you very sensitive to the perception of those rhythms. And that's why I think in general, it's much easier to derive pleasure from genres that you're already familiar with, and it's relatively more difficult to derive pleasure from a genre that's very different from your own and for which you don't have a good internal basis to generate those predictions.
Thank you for your interesting talk. I have 2 parts of a question. Maybe I have missed it. Sorry about this, but this asymmetry that you see in nucleus accumbens, you see an asymmetry, right?
Between the right and the left, you mean?
Yes. Yeah.
Yes.
Is this related to the asymmetry seen in dopamine receptors?
Well, we think, I mean, we don't know for sure. It is true that we see a relative asymmetry in many experiments, but not all. We see a stronger response in the right striatum than in the left. However, my thinking about this is that it comes from the cortical input. So one thing I didn't talk about, but we've done a lot of research on this topic over the years, is that the right cerebral hemisphere is particularly good at processing certain musical features, especially pitch-based or spectral-based or harmony-based types of features. And we see this very consistently, that the right auditory cortex, for example, I did show you in the very first slide, if you remember, with the frequency following response, we had a much stronger response on the right auditory cortex than the left. And we see that very consistently in lots of studies. So according to the model, the inputs to the reward system come from the cortical pathways. And so if the right cortical pathway is more specialized for these pitch-based or harmony-based kinds of processes, then that might explain, I think, why we tend to see more responses in the reward system on the right side, because the inputs are actually coming from the cortical part of the right side. Now, of course, music also involves other features, which maybe are more bilateral, and that's probably why we often do see bilateral responses in the striatum, but stronger on the right than on the left.
And the second part is, if I'm not wrong, nucleus accumbens is also related to impulsivity. So do you think that might be like a influence on less impulsivity, for example, or more, whatever?
Well, yeah, I don't know. I mean, the reward system is involved with many different functions. They're all kind of interrelated. I don't really know much about impulsivity. I do know that it's closely related to motivational kinds of processes. So yeah, I don't think I can really directly answer your question. I guess I would simply say that, you know, the reward system is not just for hedonic responses. It does have many other functions, and really what I'm focusing on is the hedonic part.
Hi, thank you so much for your talk. It was very interesting. So I have a question related to what has been said already a little bit about given the individual differences in music taste. So for example, some people may enjoy Latin music more, other classical music or techno music. Have you considered or have you conducted this type of experiments with different types of music? And would you expect to see differences in the type of like reward system or like the activation of the reward system, depending on how much they like the music they have? And then for the type of people that like don't show any activation or that don't seem to be that responsive to music in general, could you expect even differences in them depending on the music they're exposed to?
Yeah, so this is a very interesting question. So many years ago, the very first study we did on music reward with Ann Blood in the year 2000, we published it and we did the experiment in 1998 or 99. This was one of the first experimental problems that we faced, because we didn't know how to go about this. And it was funny because Anne, who was really running the experiment, she was actually a drummer in a heavy metal band, and I'm an organist, and I like 17th century music. So when we were talking, we said, "Oh, this is impossible. We can't do this experiment because look at the music that you like and that I like." And then we realized, oh, but when you listen to your music, you have strong emotional response and you have chills. And when I listen to my music, I also have the same response. So therefore, it's not really the music itself, because that depends on your exposure and cultural factors and training and so on. What's really important is the physiological and neurological hedonic response that you get to the music. So in those experiments, what we did was we selected for each participant the music that they said, "Oh, I really love this music." And so this way we controlled for that factor. So, one person liked classical, another one liked jazz, another one liked tango, another one liked bagpipes, Scottish bagpipes, which everyone else hates, but Scottish people like it. So, a lot depends on the exposure, but regardless of the nature of the music, the style or the genre, the physiological response is essentially identical. And there's one more little detail I can tell you about, which is that in work that other people have done, like Carol Krumhansel, for example, she's shown that genre preference is strongly tied to the music that you liked during your adolescence. So the music that you were exposed to that was important to you during adolescence is usually the music that you will like for the rest of your life. And what's really interesting about that observation is that if you look at the dopaminergic system, you see a peak in dopamine expression in puberty. So starting around age 12, 13 and peaking and then going down by the time you get to be 18 or so, there's this big peak in dopaminergic response in the striatum. So this makes a lot of sense, because it means that during that time, your reward system is kind of hyperactive. This is why all kinds of things happen during adolescence, right? You fall in love, you usually have your first sexual experience, you have a lot of exploratory behavior, you sometimes have risk-taking, which is also part of exploration, and the music that you're listening to becomes a very important part of your life, and also has to do with social bonding and separating from the parents and bonding with your peer group. All of these functions are linked to the reward system in one way or another. And so it makes sense that during this time of life, when your reward system is kind of hyperactive, the music that you're exposed to really becomes the music that that's very meaningful to you.
Hi, a very, very interesting talk. Thank you very much.
You're welcome.
I thought it was very interesting the dissociation that you found between the not ability to experience pleasure from music from other stimuli like sex or others. But I wonder whether that can be generalized to other types of sensory modalities, like those people that are not so likely to experience pleasure from music. Does it happen also with visual art, for instance, in those same people?
No, it doesn't actually. And again, Ernest can tell you more about this because he did a nice study on this. But people with music anhedonia, we call it specific music anhedonia for a reason, because we really think it's specific. Indeed, when asked to give ratings of how much pleasure they get from art, it's indistinguishable from that of everybody else. And I have even spoken to a few people with musical anhedonia who seem to really be especially interested in visual art. So there might be some kind of compensation there because they don't get so much pleasure from music. They're not so interested in it, but they do have sensitivity to visual art. So maybe it's almost a compensation. I don't know. That's only based on a casual observation. But coming back to the model, we think the reason for the music anhedonia is this relatively reduced connectivity, specifically between auditory structures and the reward system. We have no evidence for reduction of connections from, for example, the visual system into the reward system. So it does make sense in terms of the model that we have developed.
Very interesting. Thank you very much.
You're very welcome.
Okay. Okay. We are going to thank Robert. Muchas gracias. Muchas.
Gracias a ustedes, gracias por las preguntas muy interesantes y espero que el resto del simposio también que les dé mucho placer.
Gracias. Okay, now we are going to listen to Maria Benitez. She specializes in musical training as a modulator of cognitive functions in children. She's a PhD student in psychology at the Universidad Nacional de Córdoba and a PhD fellow from CONICET. Sí. She's a member of the Laboratorio Interdisciplinario de Neurociencia Cognitiva en la Universidad de Palermo, at the Palermo University. Her talk entitles Cognitive Benefit of Music Training.
Hola, ¿cómo están? Ahí va. Bueno, buen día. Thank you very much for the introduction and thank you all for being here this morning. I am very, very pleased to participate of this symposium. So I am going to present you with a work entitled Cognitive Benefits of Musical Training, carried out by the Laboratorio Interdisciplinario de Neurociencia Cognitiva, directed by Dr. Nadia Justel. So the first years of life are extraordinarily important for all aspects of development. And this is because early experiences have lasting effects. The issue of how musical experiences affects development is a matter of interest that has increased in recent years in our country. Previous research has found that musical training affects development and that human beings are born with a predisposition to perceive and to participate in musical experiences. Musical training stimulates the synaptic plasticity observed in anatomical, functional, and behavioral differences. That is, it has consequences on the anatomical and functional organization of the brain. This shows that there is cerebral plasticity, the product of adaptive processes as a result of environmental stimulation, in this particular case, musical training. So the effect that musical training has on other cognitive domains is called transference. This can be near or far as it related more or less with musical skills. So an example of near transfer is with children who, being trained in piano, receive benefits in areas such as fine motor skills. But when we talk about far transfer, we refer to children who receive benefits in areas such as memory or language that are not directly related to musical skills. So all these changes are more evident the earlier the training starts, and the longer the children are stimulated, the greater the observed changes. So here today, we are going to think of musical training as a modulator of cognitive function, and we are going to focus on memory. Memory is a cognitive function that has a process of codification, consolidation, storage, and recovery. As we know, there are several types of memory: semantic memory, episode memory. But we will focus on two of them, the emotional and the neutral memory, through a task based on the International Affective Picture System. So previous research has given an account of the influence of the age when training starts and the time of exposure to the stimulus. Now, our question was, will there be differences according to the type of training? That's why we have designed two types of training: a receptive training, in which children auditorily perceive musical sound stimuli, and an active training, where children will not only hear sound or musical stimuli, but also will produce music through musical instruments or their voices. So for all these questions, we set ourselves the objective of assessing the effect of an active or receptive musical training during 4 or 10 weeks on emotional and neutral memory in children of 4 and 5 years of age. In the two studies that we are going to present, 180 children of 4 and 5 years of age participate. They belong to kindergartens of Buenos Aires, acá in Argentina. So the first study, the children received receptive or active training for 4 weeks. In the second one, the children received the same types of training, but for a period of 10 weeks. How was the procedure we followed? Once we had the support of the institution that collaborated with the studies, we had a meeting with the children's parents. The procedure that we were going to follow with the children was explained to their parents, and they were shown the picture that the children were going to observe. The parents who agreed voluntarily signed an informed consent, and subsequently, in study 1, these children began with the training. And the end of this training, they were evaluated in their memory through a memory task. The difference with study 2, in addition to the time of exposure to the stimuli, was that in this study, we established a baseline. That is to say that the children were evaluated before and after the training. As we have seen, The memory task was carried out through the International Affective Picture System, which is a conglomerate of pictures with valence and arousal. From this set, suitable images for children were selected, images with positive, neutral, and negative content. First, the children evaluate their valence through these miles, and then the emotionality, that is how positive, for example, was a picture, through this little face. Finally, the children were evaluated on their immediate and deferred memory. That is how many images the children remembered immediately after having seen them for the first time and about 7 days later. So what were the results of our studies? First of all, we will observe the result of a first study with 4 weeks of exposure to the 2 types of musical training. And we see here the number of pictures that the children remembered immediately after having seen them for the first time. On this axis, we see the type of images observed: neutral, positive, and negative. On this axis, we can see the amount of pictures that the children remembered. In the columns are represented the 4 groups: the group of 5-year-olds, 4-year-olds with receptive training, the group of 4-year-olds with active training, the group of 5-year-olds with receptive training, and the group of 5-year-olds with active training. And we are going to focus on the immediate memory of positive images, where the group of children with active training remember more positive images than the children with receptive training. But what happened 7 days later? And we are going to focus here in the memory, deferred memory of negative images, where the children with 5-year-old with active training remember more negative images than the children with receptive training. What happened with after 10 weeks training? And we will see here in each graph are represented the types of images: neutral, positive, and negative. On this axis are represented the 4 groups, and here the amount of pictures that the children remember. In the columns, we can see the images in the baseline and after the training. So we can see here that the children with receptive training, both age, remember more neutral images before 10 weeks of training. But in the deferred pre-recall, we can see that the children with receptive training of 4 and 5 years of age remembered more neutral images, but the children with active training remember more emotional images with positive and negative content. So what are our conclusions? In first place, the active training generated an improvement in the memory of children of 4 and 5 years of age in comparison with the receptive one. 10 weeks of training were more effective than 4 weeks, and the higher results were observed in the deferred scores. So we can see here that there is an interaction between a certain types of training and a specific type of memory. Finally, lastly, I would like to thank my director, Nadia and Fabio, for the mentorship of this study. Nada más, gracias. Pueden preguntar en español, Nadia lo dijo hoy y Robert también, pero sepan que están abiertas las preguntas para los que quieran preguntar en español.
Te pregunto en español. Me parece interesante que encuentres efectos diferenciales con el receptivo y el activo. Me pregunto si esa actividad necesita ser relacionada con la música o podría ser cualquier otra actividad física, digamos, asociada a la experiencia.
Totalmente, es una de nuestras posibles explicaciones y ligada con la charla de Robert anterior. Creemos que los niños y niñas que participaron de el aprender un instrumento musical tienen otras cargas o otras, activan otras áreas que están relacionadas no solamente con la escucha, sino también con la producción de música que involucra el movimiento, y eso podría estar generando un boost, un improvement, una mejora, ahí está, de la memoria emocional, totalmente, sí.
Igual, una cosita más, tenemos otro grupo, no está acá en esta presentación, pero tenemos otro grupo de control donde hacen actividades con profesores, con los chicos, tienen la misma actividad, los pibes van, vienen, y es una actividad control que no está acá porque Mari quería poner las cosas publicadas, pero está hecho y las diferencias son iguales que las que están mostradas acá. Pero sí, está el grupo control donde hay una actividad que los pibes hacen con las mismas maestras, o sea, con todo el procedimiento, digamos. Siempre en grupo.
Totalmente, siempre en grupo en este caso. Pero sí, corrimos después de las 10 semanas, uno de 12 semanas, donde agregamos el grupo control, que es lo que le está contando Nadia. Un grupo control donde hacían una actividad de dibujo para justamente descartar esto. Y los resultados fueron, no hay diferencias pre-post entre el grupo control que dibujaba. Sí hay una diferencia en la memoria neutra para el grupo con entrenamiento receptivo, y el entrenamiento activo presentó una mejora en lo que tiene que ver con memoria emocional.
Ok. Gracias, Mari.
Gracias.
Okay, our next speaker is Verónica Díaz Abraham. She holds a PhD in neuroscience from the Universidad Nacional de Córdoba and is an Argentinian music therapist. She's a CONICET researcher and works in the Laboratorio Interdisciplinario de Neurociencia Cognitiva. She's going to present us music-based intervention, experimental studies about memory modulation.
Good morning. Thank you, Nadia, for this introduction. And thank you, Sam, for organizing this meeting and invite us to the symposium about music and neuroscience. So I have to learn how to use that. La diapositiva anterior, por favor. Gracias. So the work that I'm going to present is about the effect of music-based intervention on memory, in particular memory consolidation. I like to show the results of several studies that we run in LINC, Laboratorio Interdisciplinario de Neurociencia Cognitiva, with Nadia Justel, Morena Lopez, and other colleagues. So to start, yes, Music is a complex activity with great cognitive demand. As we learned from previous presentation, there is a strong relationship between music and cognitive function, especially emotion. And the neurocognitive mechanisms underlying music allows us to use music with as a music for cognitive modulation. Sorry. From this perspective, we call music-based intervention to the musical proposal used for therapeutic, educational, and cognitive modulation purpose. In general, this kind of activity can be implemented before, during, or after a general cognitive task in order to modify the cognitive performance of the participants. And if the musical activities that involve music perception or sound perception are called receptive intervention, and those who imply music making, music exploration, improvisation, among others, are called active intervention. Some studies suggest that this kind of intervention can be modulate attention, language, executive functions, and other cognitive functions. Specifically, some research are focalizing on learning and memory. And this is one of the main points of my presentation, the modulation of memory through music-based intervention. So as a cognitive function, memory has several phases ranging from acquisition of information, consolidation of it, recovery, and finally reconsolidation. One important point is that memory can be modulated, which means that it could be improved or deteriorated through different kind of treatment, and music has been tested in this sense. One of the most researched strategy is the use of music perception during a cognitive task. Music perception pursues different kind of results depending on the type of music used. For example, if you use arousing or relaxing music, music with or without lyrics, yield different kind of results. One of the first studies conducted by Nadia and Wanda Rubinstein employ arousing music and relaxing music after a visual learning task. In this case, they found that people who listen arousing music remember more information than people who listen relaxing music. However, we didn't find the same result when participants listen music before a visual learning task. In this case, with Morena Lopez, we employ arousing music, and we also include the music preferences of the participants. In this experiment, we didn't find difference between both interventions. So in this context, we start to think about the effect of other musical activities of the music-based intervention. And some evidence shows that music production could be more effective than receptive treatment for cognitive modulation and especially memory modulation. So with Nadia and Fayashifres, we were interested in work with musical proposal accessible to the whole population, whether musicians or not, were children or adults and so on. So for this reason, we work with a music therapy perspective about this kind of music-based intervention. Not yet, todavía el video no. Thank you. We have three conditions. Music improvisation is our experimental condition, rhythmic imitation, and bass control condition. In the music improvisation condition, the group of subjects listen this rhythm bass. This rhythm bass. We repeat this rhythm for 3 minutes. And when they want, they start to create sounds or music using this musical instrument. They can also use their voices or the body for 3 minutes. it was a free improvisation. Maybe if you wonder what is a free improvisation from a music therapy perspective, I like to show a brief example. Sí, ahora sí el video, por favor. We have to change. In this video, we can hear the background, the rhythm background, the rhythm bass in the background, and we can see the group of women playing instrument and dancing around the space. Like a ritual. Está bien, ya podemos sacar el video, gracias. Bueno, vamos a volver a escuchar, vamos a volver a ver el video. Si se quedaron con las ganas. La diapositiva anterior, ¿cuál? Gracias. Gracias. So this was a music improvisation condition. In the rhythmic imitation condition, the participant listen the same rhythm bass, and when they want, they start to imitate the pattern for 3 minutes, using musical instrument, their voice, or their body, also for 3 minutes. And finally, we have a rest condition. It's a passive control condition. In this case, the group associate remain in silence for 3 minutes. We ask them not to talk, write, or use cell phone. About the procedure of our study, each study involves 2 sessions. The first session has 3 phases. The first one, acquisition phase, the participants observed 36 images, and they had to evaluate the balance and arousal for each image. Since we work, like Maria presentation, with emotional memory, we work with the International Affective Picture System. After that, the subjects were exposed to one of the experimental conditions: music improvisation, rhythmic imitation, or rest condition. We expect that these kind of interventions, especially music-based intervention, modulate the consolidation of the first learning task. And finally, we evaluate the memory by conducting 2 tasks: recall and recognition. No, it's al revés. The second session, 7 days later from the first session, we have the different evaluation, and we conducted both tasks to evaluate memory, recall, and recognition. So with some differences, we use this procedure in several studies with children, young people, and we have a special study with Alzheimer's disease patients. So in this presentation, I like to compare the result about immediate pre-recall and deferred pre-recall. So about the study with young population, in this plot, we can see the results of immediate pre-recall. And we can see the 3 types of images: neutral, positive, and negative. We can also see the number of image remembered for each experimental condition. In white bar, music improvisation; in gray bar, rhythmic imitation; and in rest condition, in green bar. So in this case, we can see that people exposed to rhythmic imitation remember less emotional, positive, and negative images than did the other two conditions. 7 days later, we have the first recall with the same plot with the same logic, and in this case, we can see that people exposed to music improvisation remember more images in general, neutral and emotional, than did the other two conditions. When we work with older adults, we obtain comparable results in negative images. And here we can see that people exposed to rhythmic imitation remember fewer negative images than did the other two conditions. And the same result we obtain in the fair measure. But here we can see that music improvisation differ from the other two conditions. As we tell you, we run a special study with Alzheimer's disease patients. In this case, we only use music improvisation and rest condition because rhythmic imitation was considered too difficult for this patient. They had a moderate cognitive impairment. And we can also use only positive and neutral images to not generate discomfort in this patient. And in immediate measure, we didn't find a significant difference, but the patient exposed to music improvisation remember more positive images than the rest condition in the first measure. And finally, we ran a study in educational field with 4 and 5 years old children. In this case, we can see that children who imitate the pattern for 3 minutes remember few images, emotionals and neutral, than did the other two conditions. And in the fair measures, we can see that children who improvise remember more positive and negative images than did the other two conditions. So across the study, we found the following pattern or result in general. In immediate measure, we found a similar effect between music improvisation and rest condition with a lower memory performance in the adults and children who imitate the rhythmic pattern for 3 minutes. At the beginning, this was an unexpected result for us, but we think that the nature of this task, rhythmic imitation, generates a high cognitive demands, especially attentional demands, and this could have disrupt or compete with the consolidation process. The most important result for us, we can see in the first measures, because we noted that music improvisation different from the other two conditions, especially in emotional information, in positive and negative images. And we think that music improvisation functions as a second task that reinforces a previous learning task in line with behavioral tagging theory. The music improvisation effects on memory consolidation could be mediated through emotional induction. There are several studies that show that musical activities shows mental activation during musical activities, especially group productions. And we know that emotions would be modulate memory through amygdala activation. So we thought the effect of music improvisation in this sense. To end, taking into account our results, now with Cecilia Forcato and our team, we want to evaluate the effect of music-based intervention, perception, and music improvisation on the reconsolidation phase. And in parallel, we conducted a study with local music. The previous study about music perception, we used European and classical music to induce emotional state. And in this point, we start to think about the local context of the participant. So for this reason, we start a study, evaluate the effect of Latin American songs in emotion. Our intention is to use these songs as a music perception intervention. So we will focus on that in the next experiment. So this is all that I want to share with you. Thank you very much for your attention. Gracias.
¿Alguna pregunta que quieran hacerle a Vero? Bueno, ¿no?
I wonder how to implement the interventions to make value of these benefits that you see. How do you plan the recommendations to?
Perdón, no te estoy
Sí,
escuchando.
Lo que digo es, ¿cómo se traslada a la práctica las condiciones para lograr estos beneficios que vos ves?
Ujú.
¿Son recomendaciones de no escuches música en este horario, sí en este otro,
Mm.
solo antes de? ¿Cómo te lo
Mm.
imaginas implementado?
Bueno, una de las... I like to respond in Spanish. Una de las cosas que nos preocupa es la no generalización de resultados, ¿no? Porque estos estudios están realizados en un contexto experimental, entonces me parece que hay entre estos estudios y cuestiones más aplicadas, hay una serie de estudios que deberíamos realizar. Hay un montón de factores que podrían determinar también esto de recomendar, ¿no? Implementar una actividad para mejorar los mecanismos de aprendizaje y memoria. Muchas de las cosas estaban relacionadas con lo que le preguntaban anteriormente a Roberto sobre las preferencias, sobre los gustos, sobre el contexto. En estos casos se testearon algunas piezas que en general en la literatura están testeadas, pero la realidad es que hay que seguir investigando como para pensar en la parte aplicada. Sin embargo, es algo que se puede observar, que se puede aplicar, como uno ve los estudios y piensa enseguida en la parte aplicada, especialmente en el contexto educativo, ¿no? Cuando hemos presentado estos estudios en contextos educativos, los docentes y eso nos han preguntado cómo implementarlos. Y siguiendo bajo el mismo procedimiento, se podrían implementar algunas tareas de percepción musical o de producción musical luego de algunos aprendizajes. Pero me parece que sería súper importante empezar a evaluar, por ejemplo, algunas memorias más relacionadas con los contextos de aplicación. ¿Respondo a tu pregunta? ¿Continuamos?
Vamos con la...
Vamos.
Gracias, Vero.
No, por favor.
Okay, our last speaker is Ernest Mas Herrero. No lo voy a decir dos veces. Ernest, are you there? Yes.
Yes, here I am.
Okay. He received a bachelor degree in biology from the Autonomous University of Barcelona and a PhD in psychology from the University of Barcelona. During his PhD, he identified and investigated musical anhedonia. After completing his PhD, he conducted a postdoc at the Montreal Neurological Institute with Professor Satorre. He launched his own lab in the Institute of Neuroscience of the University of Barcelona. The title of his talk is Now You Like It, Now You Don't, Exploring the Brain Generators of Music-Induced Pleasure.
Okay, can you see my slides?
No.
No? Okay. Check.
Ahora sí.
See?
Sí.
All right. Well, thank you very much for all of you that are there attending this symposium. I cannot see if there is anyone. I hope that there are some people in there. And also thank you, Nadia, for organizing this very nice symposium and for inviting me. So I'm pretty sure that Robert already convinced you before that music reward relies on the interaction between two main systems. On the one hand, a cortical system involved in auditory cognition and in temporal predictions, involving the superior temporal gyrus and inferior frontal gyrus, and on the other hand, a subcortical reward system that involving particularly striatal circuits and more specifically involving the nucleus accumbens. So we wanted to test this hypothesis using a meta-analytic approach. So basically, we perform a meta-analysis of the studies, meta-analysis of neuroimaging studies, investigating musical reward. So here we start searching in the literature for previous studies using fMRI, in which participants listen to music inside the scanner, in which participants also provide ratings of any sort about liking, pleasure, and musical preferences. And they include an fMRI contrast comparing pleasant music and non-pleasant or unpleasant music. So we basically found 17 studies that met our inclusion and exclusion criteria. And the result of this meta-analysis, it's basically a nice summary of what Robert presented. So on the one hand, we found the music reward, regardless of the research team that conducted the experiment, regardless of the methods used to analyze the fMRI data or the paradigms used, we found that music reward was consistently associated with engagement of cortical brain regions here, particularly the superior temporal gyrus, including the auditory cortex, and the inferior frontal gyrus. These two structures, as Robert said, play a key role in music cognition and in generating auditory expectations and predictions. In addition, we can see how the extent of this cluster is larger on the right hemisphere as compared to the left hemisphere, further supporting this idea of this right hemispheric specialization of music perception. In addition, music reward was also associated, of course, with the engagement of reward-related structures, particularly the ventromedial prefrontal cortex and orbitofrontal cortex that Robert already pointed out its relevance in music reward, but also the striatum and different parts of the striatum actually, either dorsal sections such as the caudate, but also ventral parts of the striatum, including the nucleus accumbens. So what these results tell us, it's basically that when people are listening to music and they are experiencing pleasure inside the scanner, all these brain regions are activated, all right? However, we cannot disentangle with neuroimaging techniques to what extent this set of brain regions are actually causing musical pleasure and music reward experiences from those brain regions that are just responding as a consequence of the pleasure. So we cannot disentangle whether they are the cause or the consequence of that pleasure. So if we want to provide causal evidences, we need to somehow manipulate brain function of these regions and then investigate to what extent that actually cause changes in music reward. So brain stimulation technique, it's a quite a standard procedure in many animal research labs, and they can be very precise about the type of manipulation that they perform. They can be also very precise in terms of the location that it's being stimulated. However, the challenge here is that the best animal model to investigate musical reward is actually humans. So for obvious ethical reasons, we cannot just go and implant an electrode to targeting the nucleus accumbens in a healthy volunteer. However, we do have methods that allow us to stimulate the brain in a non-invasive way. And probably one of the most well-known techniques is transcranial magnetic stimulation, which for those that are not familiar with this method, it's basically two large magnets that generate a magnetic pulse that will go through the scalp until it reaches a targeted region, and there it will generate an electric current that may depolarize or hyperpolarize the neurons in there, leading to excitation or inhibition of the targeted region, depending in part on the type of TMS protocol that you use. We have TMS protocols, excitatory TMS protocols, and we also have inhibitory TMS protocols. However, the main limitation of TMS is that it has a limited range of action. We can only stimulate cortical brain regions. So there is no way to directly target subcortical structures, such for instance, the striatum, using TMS. Yet we can do it indirectly. How is that? Well, basically because the striatum, as any other region in the brain, doesn't work in isolation. It's highly interconnected with the rest of the brain. And particularly the human striatum, it's highly interconnected with prefrontal cortex in a way that precisely because of these anatomical and functional connections between prefrontal cortex and the striatum, it's possible to indirectly modulate striatal function by applying TMS over certain cortical regions. Specifically, Strafella and colleagues have shown in a study that has been replicated in other studies and also has been replicated in rodents, have shown that TMS over the left DLPFC, the left dorsolateral prefrontal cortex, may modulate striatal function and may even induce dopaminergic release in the striatum. So taking advantage of this method through which we can indirectly modulate striatal function, we wanted to test the causal contribution of striatal circuitries in music reward. So basically, to what extent, by applying TMS over the left DLPFC, we could modulate music reward related responses by indirect changes in the striatum. So to achieve this goal, we basically tested 17 participants that came into the lab in 3 separate sessions. On each session, they received either we apply either inhibitory TMS protocol called continuous theta burst stimulation, CTBS, or an excitatory protocol that would be the intermittent theta burst stimulation, ITBS, or sham, in which we basically would make the participant believe that we were about to apply TMS, but in the last second we would just flip the coil so that no brain was being stimulated. These protocols are pretty fast, so generally CTBS and ITBS last for about 40 seconds to 3 minutes. So basically what we would do would be stimulate the participants and then test them in a music reward task when they were under the effect of this TMS stimulation. So that when they perform the music reward task, no TMS was occurring. Okay, it was just prior to the performance of the task to test participants under these TMS effects that generally last between 20 to 40 minutes. So all the lesions that you cause with TMS is just temporary. Okay, and so after the stimulation, which was different on each day that they came into the lab, and it was randomized across participants, they listened to 45 seconds, to fragments of 45 seconds of 15 musical pieces that included their 5 favorite songs. It's a procedure very commonly used in the studies of musical reward, as Robert said before in the questions, that allow us to maximize the pleasure of each participant. And in addition, they also listened to 10 songs that we selected for the experiment, okay, and that the participant could not anticipate which ones they would listen to, okay? So participants on each of the 3 sessions listened to their 5 favorite songs and also 10 songs that differ from one session to the other. The task that participants had to do was very simple. They had to indicate in real time the degree of pleasure that they were experiencing. And for that, we were using also a very common and standardized procedure in which basically participants have 4 buttons available. One button, they have to press one button in case they are experiencing no pleasure, and another one if they experience low pleasure, another one if they're experiencing high pleasure, a 4th button finally they only have to press that button if they experience a chill or a goosebump, which will be considered sort of the musical orgasms, okay? And that's what they did. That allowed us to have a measure of their subjective reports of pleasure, but we also acquire skin conductance. So we also acquire objective measures of affect, such as skin conductance, which is an involuntary physiological reaction. We know that skin conductance actually scale linearly, or the amplitude of a skin conductance scales linearly with the amount of pleasure that people are experiencing. Particularly when people are experiencing a chill, we see a huge rise of the skin conductance response amplitude. And it's also related to individual differences in music reward sensitivity, as Robert showed before. And it also reflects, in general, the intensity of the hedonic experience that participants are experiencing while listening to music. So that way we had like objective markers that were independent of any conscious evaluation, but also subjective reports of pleasure, which also relates to this more conscious processing of that experience. In addition, participants also had the opportunity to purchase our music selection. And here we use an auction paradigm similar to the one that Robert also presented. Before, participants had the opportunity to purchase for each of the musical items at the end of each excerpt, and they had to indicate how much money they were willing to pay to buy each specific musical item. That would allow us to have a sort of a measure of how motivated they were to listen to music, to listen to our music again. So overall, we end up with 3 variables of interest: objective markers of effect, as measured by subjective, by physiological reactions, as skin conductance. We also had measures of subjective pleasure by their real-time ratings, and finally a measure of their motivation to listen to music again, as reflected by the amount of money that we were willing to pay. So that way we had like different tiny related processes, yet still independent processes related to reward processing. And this is like quite a summary of the results that we got. So here in blue, you can see represented changes with respect to sham. So we use always sham as our baseline, following excitatory stimulation, intermittent theta burst stimulation. And in red, what you have represented here is changes following continuous theta burst stimulation, following the inhibitory protocol. And basically what we found was that following excitatory stimulation, participants were more likely to enjoy the music, as reflected by their subjective reports of pleasure. We also found a greater amplitude of the electrodermal activity or the skin conductance response, and also participants were more willing to pay for our music selection. They were more motivated. While we found the opposite effects following CTBS, continuous theta burst stimulation, or the inhibitory protocol. Okay, all these differences here are significant. So this finding seems to indicate that the TMS procedure that we use can effectively modulate both affective reactions to music, as measured by skin conductance, also subjective reports of pleasure, and finally motivational aspects of music reward. And here in this study, we basically speculate that those effects were mediated by indirect changes in the striatum. That was actually the goal of using, that's why we use this procedure. However, it's true that that was sort of a speculation, because we basically based that on previous findings. But here we didn't measure brain activity. We didn't have neuroimaging data. So it could be perfectly possible that these effects were just driven by TMS-induced changes in some other brain regions, nothing to do with the striatum or other reward-related structures. So to answer this question, what we did was a second study, a follow-up study, in which we again tested 17 new participants that did exactly the same, but now in the scanner. Well, at least the task. The stimulation was done in a room just in front of the MRI scanner, we would apply inhibitory, excitatory, or sham stimulation on each of the sessions that participants would come, and then they will jump in the MRI to listen to or to perform this music reward task, which basically consists on the same things that I mentioned before. They listen their 5 favorite songs, and also our music selection. They had to indicate the degree of pleasure that they were experiencing using this 4 buttons procedure, and they also had to, or they had the opportunity to purchase our music selection. Well, those are the behavioral results. We replicate our previous findings. That was kind of a relief, and it was also nice to see that we could replicate the same behavioral effects even in a noisier environment as it is the MRI. So in blue, again, you see basically changes with respect to sham following excitatory stimulation. In red, changes with respect to sham in continuous theta versus stimulation after applying the inhibitory protocol. So again, individuals were more likely to provide greater subjective reports of pleasure following excitatory stimulation rather than inhibitory stimulation. They were also more likely to spend more money on our music selection following excitatory than inhibitory stimulation, and they also experienced more chills and spend more time reporting chills following excitatory than inhibitory stimulation. Okay, so behavioral part done, it's consistent with our previous results. Then we move to the neural level, what's going on in there. So we basically look at brain activations at reward-related structures previously associated with music reward. Specifically, we look at brain activations at the ventromedial prefrontal cortex and also different subregions of the striatum. That is the caudate, the putamen, and the nucleus accumbens. What the first meta-analysis, the meta-analysis that I showed you at the beginning, what this meta-analysis tells us is that there is a positive correlation between pleasure and engagement of these brain structures. So we first checked that at sham, which is represented here in green, and here you can see brain activations when participants were reporting no pleasure, low pleasure, high pleasure, or when they were experiencing a chill. And we can see that indeed there is this positive correlation between pleasure and brain activation in these reward-related structures. Notably, after applying continuous theta versus stimulation, that is the inhibitory protocol, the slope of this relationship was completely flat. So no relationship at all between brain activations and pleasure. However, when we apply excitatory stimulation, we can see how the slope is even larger than what we found in sham. So it seems that these reward-related structures were responding more to pleasure in the excitatory protocol than in sham. And here you kind of have a summary of all of that. Here you see pleasure-related activations following excitatory stimulation compared to sham, pleasure-related activations following the inhibitory protocol, continuous theta versus stimulation as compared to sham for each of the 4 brain structures that we look at. These effects were not found in other control brain regions that we use as a control. So it seems that these effects were quite specific to the reward system rather than other brain regions. So that's good. It seems like we replicate our behavioral effects. And in addition, we see that those brain regions that we thought that were actually involved in these processes, are also modulated. Next, what we wanted to know was to what extent each of these brain regions were contributing to these behavioral effects. So can we explain these TMS-induced changes in motivation and pleasure based on TMS-induced changes in nucleus accumbens, caudate, putamen, or ventromedial prefrontal cortex? We then perform a series of regression and analysis on correlational analysis. And basically what we found was that among these 4 different brain structures, only TMS-induced changes in the nucleus accumbens predicted changes in pleasure and motivation. So TMS-induced changes in subjective reports of pleasure and TMS-induced changes in the amount of money that participants were willing to pay to buy our music selection. Based on these results, we can sort of conclude that the striatal pathways, and particularly the nucleus accumbens, are indispensable to experience music reward. But it was not only about the engagement or the activation of the nucleus accumbens, it was also about how these nucleus accumbens interact with the right auditory cortex, but not the left. So in kind of consistent with what Robert already showed, we found that TMS-induced changes in the connectivity strengths between the auditory cortex and the nucleus accumbens predict as well TMS-induced changes in pleasure, which further support this model that musical reward relies on the interaction between these two systems. Here, we also make another speculation. We basically speculate that all these processes were mediated by dopaminergic transmission. Why dopamine? Well, because as Robert showed, we have seen that music reward nicely correlates with dopamine release, but also because this TMS procedure has been previously shown to modulate dopaminergic transmission in the striatum. However, we didn't measure dopamine in here. We just had indirect measures of brain activation, but that's it. So to further investigate the contribution of dopaminergic transmission in musical reward, we perform a third experiment in which now we perform a pharmacological intervention in which participants again came in three separate days, and we administered either levodopa, a dopaminergic precursor, risperidone, which is a dopamine antagonist, or lactose, which is just a placebo. And these 27 participants performed the exact same task that I explained before. So we had measures of motivation, measures of subjective pleasure, and we also had objective markers of affect. And here you can see sort of a summary of the main results that we got. So basically here what you can see in green represented changes with respect to placebo following levodopa administration. In red, what you have is changes with respect to placebo following risperidone. And what our results indicate is that similar to what we found with the TMS procedure, individuals were more likely to experience chills when they were under levodopa effects than when they were under risperidone. Notably, we found the reverse effect when looking at low pleasure ratings. So individuals were more likely to report low pleasure ratings when they were under risperidone, the dopamine antagonist, than when they were under the effects of the dopaminergic precursor levodopa. In addition, participants were also willing to spend more money to buy our music selection when they were under dopamine precursor levodopa than when they were under the risperidone. Finally, we also found similar effects in relation to the skin conductance and or electrodermal activity. You see here you see it more nicely represented. In green, you see pleasure-related skin conductance responses following levodopa as compared to placebo. And in red, you can see pleasure-related skin conductance response to under risperidone as compared to placebo. So based on these results, we can conclude that music-induced pleasure relies on dopaminergic transmission. And that would be great, but the point is that these results are at odds with previous studies conducted in rodents investigating primary pleasures. So there is a large body of research investigating this topic, investigating the neural generators of pleasure in animals and using as a model sweetness, for instance. And what these studies tell us is that dopamine role, it's basically in wanting and learning. So basically the role of dopamine would be to somehow assign a value to a particular experience so that it will guide learning and our motivation. However, if we manipulate dopaminergic transmission, we will see that rats are more willing to work to get a reward. However, when we look at effective reactions, we do not see any change. So for instance, when rats are tasting something sweet, you see that they start licking their lips like crazy. Newborns also do that, and this is something shared across many other animals, and it's something quite specific to sweetness. And you see that the sweeter the liquid solution they are tasting, the more they will do that. So, however, when you manipulate dopaminergic transmission in nucleus accumbens, for instance, you do not see any change in the frequency of the rats licking or anything like that. If you want to see a change on that, what you have to target is opioidergic system. So by administrating opioidergic agonist in certain parts of the nucleus accumbens, here represented in red, you can actually increase the frequency of licking in the rat or completely reduce it, okay? In terms of dopamine, there is no effect at all. So the general idea or the model, the actual model about how these two systems interact is that on the one hand, opioidergic circuitries are the actual responsibles of the pleasure. They are the actual responsibles of painting an hedonic gloss into a sensation. But then dopaminergic circuits are those that are involved in more evaluating that experience, assigning a value and representing that value so that it will guide our behavior and it will guide learning and motivation. So what's going on here? How is it that we find not only changes in terms of motivation when we manipulate dopaminergic transmission in musical reward, but we also find that people are experiencing more chills and their physiological reactions are also greater under dopamine than when they are off dopamine. So we think that there are two potential hypotheses here. One thing would be that music reward and abstract rewards in general are just something different that relies completely on dopaminergic transmission, and opioidergic circuits has nothing to do in here. And a second hypothesis is that opioidergic circuits are still essential for these more hedonic, core hedonic reactions, those hedonic reactions that are independent of any conscious processing, any conscious evaluation, but it's just that abstract rewards requires an additional step involving dopaminergic transmission. So to test that, we perform a fourth experiment in which basically participants did again the same. This one was done with 21 participants, but now we administrated drugs, opioidergic drugs. So basically oxycodone, which is opioid agonist, naltrexone, which is an opioid antagonist, and again, lactose as a placebo. And they did exactly the same task as all the other guys. And in contrast to what we found with our TMS and dopaminergic study, we did not see any modulation, any drug modulation in subjective reports of pleasure, in number of chills, not even in the amount of money that they were willing to pay. So behavioral effects, there were no behavioral effects, basically. There were no drug effects on participants' evaluation of music. Yet we found a pretty large effect, even larger than the one that we found in the dopaminergic study, in the involuntary physiological reactions such as skin conductance. So here in green, you have represented pleasure-related skin conductance response following the dopaminergic agonist, oxycodone, and in red, you have represented pleasure-related skin conductance response following the opioidergic antagonist. So we believe that these findings are kind of mimic what previous studies in rodents have shown using primary pleasures. So basically, that opioidergic circuitry is critical for objective hedonic reactions, as well as what may happen with primary pleasures. So trying to build a model here as well on this interaction and this dissociable role of dopamine and opioids in music and primary pleasures, what we think that it's going on, to summarize, it's basically that when we're tasting something sweet or a rat is tasting something sweet, the generation of pleasure is fully mediated by opioidergic circuits. Yet after that, there is a more evaluation of that experience that may lead the rat to work to obtain that reward again, to learn from it, and also to boost their motivation. However, when it turns to music, it might be the case that it's not only about a piezoelectric circuit, but we may need, prior to that, some additional steps involving dopaminergic striatal circuitries, so that when these dopaminergic striatal circuits are enhanced, it's possible for auditory information to have access to this opioidergic system, leading to pleasure and later to motivation. And I think that would be all. I want to thank everybody that made all these projects possible, and also thank you very much, everybody, for your attention.
Gracias, Ernest. Hermosa charla. ¿Preguntas?
Thank you, Ernest. I love your talk. Robert said music pleasure is related to the reward of prediction errors. I wouldn't enjoy a pure wave or a constant rhythm that much, so that makes sense. But I wouldn't enjoy music that I can predict at all. How do you take that balance into account? And do you think you could make me like music that I'm not familiar at all with, like transcranial excitatory stimulation or levodopa?
Well, it's a good question. So what we think is that music, it sort of reflects a reward for learning, right? So in those contexts in which you cannot learn anything, either because it's fully predictable, there is nothing to learn, or because it's so complex that you cannot grasp anything, either there. So in most conditions, there is no learning, pleasure doesn't happen, okay? And we generally like a moderate level of complexity, which keep a good balance between predictability and surprise. That's to say something about the first part of the question. What I think that we are doing with TMS, we're not modulating or shaping your preferences towards one music or the other. I think that we're basically doing is increase the intensity of that you already like. So I believe that if we want to shape participants' preferences artificially with brain stimulation, we should trigger cortical brain regions involved in music predictions and anticipation, as for instance, the inferior frontal gyrus. So we should trigger more this cortical system that Robert was talking about, involved in auditory expectations. And maybe then we could shift your preference for maybe music more complex that would you generally like, or music simpler of what you generally like. I don't know if that answered your question.
Thanks. I have a question that I am not sure I am going to phrase it correctly, but when Robert and you and the rest of the speakers refer to pleasure, the way you relate to it in one way is the pleasure we feel with food or with drinking or with drugs, and all those relate to primary needs of animals. Yeah. Now, music and visual arts have the aspect of aesthetic pleasure, placer estético, and that's not feeding any basic need of any animal. And so I wonder if in your studies you could compare the factor of pleasure through sound with, let's say, curiosity. You ask a person, tell me what would you like to know, and then you, through words, you satisfy that need. Would that be equivalent to music in some way or another, or music that doesn't really produce pleasure but just a familiarity? Is it really always aesthetical pleasure related to music? And beyond that, is aesthetical pleasure related to a just evolution of pleasure, or is it really an emergent property of the brain where it's not really related to that basic idea of pleasure as we know it?
Yeah. So what I think is, as I was saying before, that music reflects sort of a reward for learning. And that has a clear evolutionary advantage. You know, if you're good at learning and you can learn what's going on in the environment, you will be able to better adapt to that environment, particularly if it's a changing environment. So as you said, I think it's tightly related to aspects such as curiosity, you know, and the need that we have to get new information and to face new challenges. And so basically what I think is that it's kind of information as a reward. It's something that it's not only unique of humans. Monkeys, for instance, also show a lot of curiosity. fMRI studies and electrophysiological studies have shown that all these reward-related structures also relate or are engaged when new information that is relevant to understand the environment is provided. And I think that in our case, because we have more probably frontal regions that has provided us with a better way or better mechanisms or more efficient mechanisms to learn, to understand, to reason, that has led us to extend the range of activities that we might find pleasurable. So we may face more challenges and more challenging than other species. And I think that music kind of reflects that. Music is a perfect stimuli to generate patterns and to play with things like predictions, expectations, and surprise. And I think that much of music's powers come from its ability to generate all these expectations and our need to get new information constantly. So I think it's a reflect of an evolutionary advantage that is to find learning or to attach a reward to learning. Yeah, I don't know if that kind of answered your question.
¿Alguna otra pregunta? Bueno, entonces damos por concluido el simposio. Muchas gracias, Ernest. Mil
Nada,
gracias.
vosotros.
Un aviso.